There are 4 steps in aerobic respiration. Glycolysis, pyruvate oxidation are the linkage reaction, the Krebs cycle, and the ETC. Glycolysis means sugar splitting. The 6-carbon molecule glucose turns into 2 3-carbon pyruvates through a series of steps. Here you can see the products, and here you can see a detailed explanation of the 9 steps. ATP that is produced during glycolysis is produced via substrate-level phosphorylation. This results when an enzyme pushes a substrate with the phosphate group with an ADP molecule together so that the phosphate transferred to the ADP, creating ATP. Keep in mind that there is only a 2 net gain of ATP because glycolysis uses 2 ATP during its steps. In the linkage reaction before the Krebs cycle, the 3-carbon molecule pyruvate is converted to a 2-carbon molecule added to a coenzyme A, forming acetyl-CoA. In the Krebs cycle, the 2-carbon molecule acetyl-CoA is added to OAA, a 4-carbon molecule resulting in 6 carbons. The next step results in a 5-carbon molecule releasing CO2 as one of the carbons. Then another step reduces it to 4 carbons. Once it is at 4 carbons, it then turns into a series of different isomers and creates these products along the way. The Krebs cycle undergoes 2 cycles per glucose molecule because it has 2 pyruvates per glucose. These molecules here are electron carriers and will take the electrons to the electron transport chain. The electron transport chain is located in the inner mitochondrial membrane. As the name suggests, electrons are moved from one part of the chain to the end of the chain. The chain consists of protein electron carriers and coenzymes. The carriers can only hold full hydrogen atoms, while the coenzymes can only hold electrons. This results in hydrogen ions moving from one side of the membrane into the protein carriers out the other side of the membrane when they reach the coenzymes. Over time, this will result in a higher concentration of hydrogen ions on one side of the membrane than the other. The lipid bilayer is impermeable to hydrogen ions, so the high concentration of hydrogen ions must move down the gradient through a special protein called ATP synthase. When the hydrogen ions move down their concentration through facilitated diffusion and ATP synthase, they will turn a turbine which puts on a phosphate group onto ADP, resulting in adenosine triphosphate, otherwise known as ATP, the energy molecule for life.